Hi, can everybody hear me okay? Please let me know if you can't at any point. So hopefully the talks that Dan and I are going to give this evening should be quite complementary. So my background is molecular biology. I converted to neuroscience later on, and I've been working in the Department of Psychiatry for around 10 years now. And my interest is really very much in understanding how genetic factors influence whole brain function. And then I suspect Dan is going to talk more about how psychological processes relate to what we see in changes in mental illness. So I'm going to focus on schizophrenia tonight, but much of what I'm going to say is generally true for the psychiatric illnesses. So I thought I'd better start by talking about what schizophrenia is, because it's something I find can be quite misunderstood. So typically when people think of schizophrenia, they think of what we would call the positive symptoms. This is the psychosis that patients suffer from. So these are things like hallucinations, delusional beliefs, and so on. In addition, though, patients also suffer from a cluster of what's called negative symptoms, and these include things like apathy and withdrawal from, for example, social interaction. And as well as this, it's not part of the diagnostic criteria, but patients also suffer from a range of impairments in what we call cognition, that is, things like attention, decision-making, and working memory. So at the moment, we have drugs, they're not perfect, but we do have drugs that somewhat treat the positive symptoms, so stop people from being acutely psychotic. But what we don't have really any good treatments for at the moment are the negative symptoms and the cognitive impairments that patients suffer from. And there's a lot of interest in developing better treatments for these aspects of the illness, because we think that one of the problems that patients have in the long term is really reintegrating into society, holding down a job, paying mortgage and things like that. And it's actually the problems with cognitive impairments particularly that stop people from doing these things successfully. So what my research has been interested in particularly is whether we can improve cognition to help patients with schizophrenia. So schizophrenia is what's called a complex disorder, which sounds like kind of a fatuous term, but it's something that has quite a specific meaning, namely that it's a disorder that is caused by multiple genetic factors and multiple environmental factors that interact with one another. So there's no one gene for schizophrenia, nor is there one environmental factor that will cause schizophrenia. The way I tend to think about it is that genes influence the brain that you have, and that how you respond in the long term depends on the environment in which you encounter. So if you like, genes can make you more or less resilient to environmental adversity of one kind or another. This is how I tend to think about schizophrenia. So I think it's a really, really exciting time to be in this field, because in the last few years we've really started to get a good handle on what some of the genes associated with schizophrenia might be, so the specific bits of DNA that seem to predispose to developing schizophrenia and other psychiatric illnesses as well. And this has really come about because we've in the last few years done really, really large-scale genetic studies in hundreds of thousands of people, comparing patients with various mental health conditions and patients who don't have these conditions. So far, this is for schizophrenia, there are different kinds of genetic variants that seem to predispose to illness. So at one end, you have what's called the common variants, and these are typically single letter changes in the genetic code that are found very commonly in the population as a whole. So everybody in this room will have one or other form of these kinds of letter changes. And each one of these can have a very, very tiny effect on your risk of developing schizophrenia. So it might increase it by maybe 1% at most. But now we've got very large sample sizes, we're able to identify these very, very small risk effects. And a paper that was published about 18 months, 2 years ago now, found 108 of these single letter changes that seem to increase risk for schizophrenia. At the other end of the spectrum, in addition to these very small changes that have a very small effect on risk and are very common in human populations, there also seem to be very large-scale changes in the genome which have a much bigger effect on risk. So I think one of the things that's been surprising to me as somebody who works in molecular biology is just how flexible the genome is, if you like. So everybody in this room will have fairly large-scale deletions or duplications of bits of DNA, and in most cases it has absolutely no effect on you whatsoever. I think that's something that's really surprising to me, and it's something that we've only really found out as we've had the technology to sequence the genomes of lots of different people. However, some bits of the genome do seem to be more critical. So there are regions that if you have a deletion in one copy of a specific region, it can increase your risk of developing schizophrenia by much greater margin, so maybe 40-fold. So these we can find, although they're rare because they have such a big effect, and there's around 10 of these big deletion or duplication regions that have been identified to date. What's more difficult to detect, these things that fall in the middle, so small things that are rare, essentially, because the number of people you have to look at is astronomically larger, and typically you would need to sequence individual people's genome, which is much more expensive. However, people have started to do this, and there is one of these regions being reported so far. It's a very small change, but it profoundly affects the function of the protein that the gene encodes. And interestingly, the gene that's implicated is one of the ones that's also been associated in some of the other genetic variants as well. So you can kind of think of this as being sort of a continuum of genetic risk, if you like. On one level, you've got very common variants that have a very, very small effect. And on the other end, you've got big variants that have a much larger effect on risk. So you'll notice that I and many other people will talk about genes for schizophrenia. But the thing to note is that it's a complete misnomer. There are no genes for schizophrenia, because that's not what genes do. Genes don't encode hallucinations, they don't encode delusions. What genes do is to encode molecules. So really, my area of interest is trying to understand how we get from these molecules up to the level of the whole brain. And I'm going to talk very, very briefly about some of the studies that we've done to interrogate this. But I think probably even just looking at it, you can see that you need a load of different kinds of technical approaches to study the brain at all of these different levels. So essentially, my group's research aims to fill that gap between genes and the level of the whole brain. So really, it aims to understand how individual genes influence brain function. And the reason for doing this is twofold. Firstly, if we have an understanding of the genes that predispose to illness, that could help us to identify new drug targets, for example. But it's also interesting from a basic science point of view in that it helps us to understand the biology of these illnesses. So what I'm going to focus on today is just a very brief example of where we've started to move from gene to whole brain function in a way that's relevant to treatment. So one of the most prevailing hypotheses in terms of the causation of schizophrenia and psychosis in particular, one of the molecules that's been best studied in psychiatry with regards to psychosis particularly is a molecule called dopamine. And this is a chemical messenger that permits communication between brain cells. And there's a region of the brain, deep within the brain, called the striatum, where it's been found fairly convincingly that if people are suffering from psychosis, you have increased dopamine signaling in that region of the brain, deep inside the brain. And it seems to track with the psychosis. So as people get better, the amount of dopamine signaling reduces. And the drugs that we have for psychosis all target this dopamine receptor. However, in addition, as well as the dopamine signaling within the striatum, dopamine is also important in other areas of the brain, particularly a region called the cortex, which is kind of here at the front of your brain, where it's important for regulating cognitive function, for example. And conversely, rather than having too much dopamine, as we see in the striatum, for various reasons we think that patients with schizophrenia have insufficient dopamine signaling in the cortex. Now, all of the drugs that we have available at the moment target a kind of receptor that's primarily found in the striatum. So they don't really do much to cortical dopamine. And even if they did treat cortical dopamine, they would have the opposite effect. So we want to boost cortical dopamine, but actually we're having insufficient dopamine in patients. I know it seems odd to have this kind of too much dopamine in one region and not enough in the other, but from animal studies, we know that there's kind of a yin-yang relationship between these two regions. So if you increase dopamine in the striatum, you get a decrease in the cortex and vice versa. So my research focuses on a gene, a particular gene, that we've been interested in for a while in psychiatry because it acts to break down dopamine. The product breaks down dopamine. So this is what I mean by the relationship between dopamine and the striatum and the cortex. So in the striatum, we think there's too much dopamine, and that's what underlies the psychosis that we see in patients. But conversely, in the cortex, we think there's insufficient dopamine in patients, and so we'd want to specifically boost cortical dopamine whilst not affecting or dampening down striatal dopamine, if that makes sense. Okay. So my research for about the last 10 years or so has focused on a particular gene called catechol-O-methyltransferase, or COMT. And this is a gene that's been of interest in psychiatry for a while because the product of the gene is an enzyme that breaks down dopamine. And one of the reasons it's been so well studied is that the human COMT gene contains in its sequence one of these single letter changes to the DNA code, and it directly affects the function of the COMT protein. So what it does is to change the activity of this enzyme that breaks down dopamine. So what you're looking at here is COMT activity measured in post-mortem brain tissue from humans, split by their what we call COMT genotype, so which form of the COMT gene they have. So people who have 2 copies of what we call the VAL form have high enzyme activity, and people who have 2 copies of the MET form have low activity, whilst people who've got one of each have activity in the middle. So this is quite a nice thing from a research point of view, because it means that we can have a very easy thing we can measure, this VAL or MET form, which we can measure genetically. That's an easy thing to do, that gives us a proxy for what's going on with dopamine function or COMT function in the brain. So over the years, I spent quite a lot of time investigating the effects of COMT at various different levels. I haven't got time to go through the details today, but what we've shown quite nicely is that COMT affects dopamine specifically in the cortex, so it doesn't seem to have any effects in the striatum. And if you remember, this is a good thing because we don't really want to mess about with striatal dopamine in patients with schizophrenia because it could exacerbate psychosis. And we've shown quite nicely that the effects of dopamine map onto effects that you can see at the network level. We can measure these with things like functional magnetic resonance imaging, for example. And again, you see fairly specific effects of COMT in these cortical regions. And we and others have been interested to see whether this translates into effects on cognitive function. So I'm just going to show you one piece of data today, which investigates the relationship between COMT, dopamine, and cognitive function. In the case of what I'm going to show you, it's something called working memory, which is the ability to remember and manipulate information in a relatively short space of time. So, say I give you a 10-digit number and ask you to repeat it backwards to me, for example. The interesting thing about dopamine in the cortex, though, is that it's a little bit like the 3 bears. In order to have optimal cognitive function, you want to have an intermediate amount of dopamine. So here on this axis, higher is better function. And if you have either too little dopamine or too much dopamine, this leads to relative impairments in performance. Now, for various reasons I haven't got time to go into today, we think that people who have this genetic variant in COMT naturally fall on different places on this so-called inverted U curve, like an upside-down U. So we think that people who have 2 copies of the VAL form of COMT are on the rising phase of the curve, whilst people who have 2 copies of the MET form are nearer the top of the curve. And this translates into relatively better performance in the MET people compared to the VAL people. So what we were interested to ask is what happens when you manipulate COMT activity using a drug. So we have a drug called tolcapone, which blocks COMT activity, so it should increase dopamine in everybody because COMT is normally chewing up dopamine. So when you block its activity, you boost dopamine levels. So you'd predict if you're a VAL person that this would be a good thing, that it should push you nearer to optimal levels. However, if you're a MET person, you'd expect maybe it would have no effect, or it could even make you worse because it could push you to excessive dopamine levels. So we decided to test this directly. The way we did this was to recruit people who either had the high activity form, the VAL form of COMT, or the low activity form, and then we gave them this COMT blocker or placebo in a double-blind study. And we looked at the effects on working memory. And what you can see in the placebo group is that, exactly as I'd shown you on the previous slide, we find that the Val-Val people perform worse on this cognitive task than the Met-Met people. In contrast, when you give them tolcapone, this relationship flips round. And you'll see I've kind of ordered them here in terms of the amount of dopamine they've got, and it looks a lot like an inverted U. So this is a really nice example, and I believe it's the first example showing a true interactive effect of a drug that affects a gene and genetic variation within that gene on cognitive function. This really nice opposite effect depending on whether you're a Val-Val or a Met-Met. So we're interested in pursuing this further by potentially using this drug in patients with schizophrenia and other patient groups that have impaired cognition, because we predict that it should be beneficial, it should boost levels in these groups. So just before I hand over to Dan, I'd like to summarise briefly. The first thing, if you take away anything today, the thing is this: there are no genes for schizophrenia. People may use that as shorthand, but it's not what genes do. Genes affect the molecules that are present in your brain, but they do affect how likely you are to develop the disorder. Some of the genes that increase risk have now been identified. I haven't shown you some of the work that we're doing in this area, but for example, we're interested in calcium signalling, and there's a group of proteins that affect calcium signalling that are very strongly implicated by these studies. So I hope I've shown you today that moving from a risk gene up to the level of whole brain, at least in terms of cognitive function, is possible. It is complicated, and it's a very collaborative field, so I work with people in all different fields of neuroscience, and it's something I enjoy very much, and it also involves patients, families and relatives and so on. And I think we will have to use multiple technical approaches, which is hopefully where I'm going to segue quite nicely into Dan next. But I think understanding these links between genes and brain have the potential to not only identify the mechanisms involved in causing these illnesses, but also have the potential to identify new treatment targets. So I'd just like to thank my lab, who are shown up here in the top, and particularly Sarah Farrell, who collected the data that I showed you earlier, along with Paul Harrison. And I'm going to hand over quickly to Dan so that we can move on.